Hi, how you guys doing? Okay, we the last time we had a lecture, the topic was cell membrane diffusion. Remember all that went on forever. And the last thing was mitosis, which is cell replication. Do you remember? Okay, so when we finished that, I said the next thing we'll talk about is meiosis, the making of the sex cell. I said that's in chapter 3, and it is it's in embryology, and the only thing you're gonna be responsible for testing purposes in that chapter is meiosis. Stuff behind me. Got it? So if you're following along in the textbook, I think it's towards the end, and it'll start talking about meiosis, and it'll talk about gonads and stuff like that. Got it? Now, the way I'm going to do it here, this is a D on your syllabus, which means directed teaching. Stuff's behind me. I'm going to be writing stuff on the board. So I'm trying something a little different so you can see the words better, but it makes the screen a little smaller. So the field of view on the screen a little bit smaller. So I'm going to go through the first column there, and I'll tell you what it is. And then I'll go through the second column and we'll discuss some of that. And then I'll move the screen so I have this clear thing over here because I'll be doing some drawings. Got it? Okay. Unfortunately, you can't ask me questions, but that's what SI is for. And Brooke will help you out there. Okay, here, meiosis. Meiosis is called, well, it's called meiosis, but it's reduction division. Two things are going on. You're reducing the number of chromosomes from 46 to 23. Remember in mitosis, mom gave you 23, dad gave you 23. Together we had 46 or 23 pair. Now what we're going to do is we're going to split those pair again into mom and dad, and then we're going to duplicate mom and duplicate dad and stick them together. So instead of having sister chromatids like we did in mitosis, we're going to have a tetrad, 4 of them stuck together, okay? And then what we're going to do is we're going to divide it into a dyad, and then we're going to divide it again into 23. So we'll have 2 sets of mom and 2 sets of moms and 2 sets of dads. Got it? Got it? So that's why we call it reduction. We're reducing the number of chromosomes, and division because we're dividing the cell twice. So it's like going through mitosis, sorta, 2 times. Remember, we went through mitosis. 1 time we got to telophase, we had 2 daughter cells. Here we're going to go through, get 2 daughter cells, and then run it through again. So that's why we end up with meiosis 1 and meiosis 2. We're good, okay? Now, meiosis is the making of the sex cell. The making of the sex cell. I'm going to move this over just a little bit so I can stand to the side. Okay, meiosis is the making of the sex cell. The sex cell, also called gamete, G-A-M-E-T-E, gamete. Okay, also we call it the sperm cell, we call it the egg, ovum, ova, all those things. They're all true. Generally speaking, the sex cell, that applies to both. Gamete applies to both. Are we good? This occurs in the gonads. Yes, women have gonads just like dudes. So if you look at my definition, the making of the sex cell happens in the gonads. The male gonads are the testes. The female gonads are the ovaries. Got it? So what I'm going to do is when I get down to the end, I'm going to go through exactly what happens in the male. I'm going to pause this, erase it all, then I'll show what happens in the female. Good. Okay. Male meiosis. Male meiosis is called spermatogenesis. Male meiosis is called spermatogenesis. Female meiosis is called oogenesis. Oogenesis. And the result of spermatogenesis is sperm cells. The result of oogenesis is an oocyte. Oocyte, egg, got it? Ovum, ova, oocyte. That's how you got the name, double O. Are we good? Now, there's some similarities between mitosis and meiosis. Remember in mitosis, we had prophase, metaphase, anaphase, telophase, and there were certain characteristics. The chromosome, the chromatin, was tightening up around the histones in prophase. Yes? The nuclear envelope was breaking up. The centrosome split apart into centrioles at the poles. Yes? That was in prophase. Metaphase, the sister chromatids lined up in the middle. M for middle. You remember that? Anaphase, they pulled apart. The spindle fibers shortened and split the centromere. And then telophase, the nuclear envelope reappeared, it furrowed until it pinched off and you had two cells. Got it? Exactly the same thing in meiosis. Prophase, metaphase, anaphase, telophase, but it goes through it 2 times. So the same mechanism is occurring. It's just happening twice. And inside, it's a little bit different. Instead of having 2 sisters together like this, and they break apart, you have the 2 sisters and 2 brothers together, and they break apart, and then they break apart again. You get it? I'm going to draw it for you. Okay. All cells in our body, except for the sex cells, all cells in our body have 46 chromatin. You got 23 from mom, 23 from dad. You have 2 pairs of 23. You got 46. Good? Good? Okay. That is said to be a diploid cell, and we abbreviate it to N. Diploid cell, we abbreviate it to N. Now, once we go through meiosis 1 and 2, prophase, metaphase, anaphase, telophase, 2 times, you don't have a diploid cell. You have a sex cell, and that's called a haploid cell, abbreviated N. So in our case in humans, N is equal to 23. 2 times N is 46. That's a diploid cell. You get it? Okay, so you need to know these terms. What is diploid? How do we abbreviate diploid? What is haploid? How do we abbreviate haploid? What is prophase, metaphase, anaphase, and telophase? What is meiosis 1 and meiosis 2? Those are all tied together in that statement, that paragraph I just gave you. Okay, the next thing I want to talk about is these guys right here. We see homologous, analogous, homologous, heterologous. You see structures and chromosomes. Chromatin, same thing. In the human reproductive system, males have certain parts, females have certain parts. But embryologically, they're the same. Yes? So when they have the same, remember me giving you this? Homologous structures, write this down, homologous structures have the same origin, O-R-I-G-I-N, may or may not have the same function. Same origin may or may not have the same function. Okay? Analogous. Now leave a space because I'm going to give you some examples. Analogous structures. Different origin, same function. Different origin, same function. Analogous. Here's where I'm at. Okay, so what does origin mean? Origin means it's made out of the same thing, it's just modified. Here's what I mean on homologous. My arm and the wing of a bird, homologous. If you took the feathers off, you took the skin off, they'd have a humerus, a radius, and ulna, and fingers. Okay, same thing for a bat. A cow. My arm and the front leg of a cow, homologous. Do you see? Okay, analogous. Okay, my arm or the wing of a bird. Now let's do this one. The wing of a bird and the wing of a butterfly. They're analogous. They're not made of the same thing, but they have the same function. They allow the organism to fly. If you take the wing of a butterfly apart, there isn't any humerus, radius, ulna, and fingers, phalanges. There is none. Get it? Okay, you're thinking, okay, now, how in the heck does that apply to human reproductive system? Okay, here you go. Get ready to write because this is going to show up on an exam. The glans penis, that's the head of the penis, and the clitoris, not clitoris, it's pronounced clitoris, are homologous. They have the same origin. A clitoris is just a small glans penis, or the other way around, a glans penis is just a large clitoris. Yeah. The prepuce, foreskin that goes over the glans penis of a male. Females have a prepuce too, and it goes over their clitoris. Wow. Keep going, Daddy, keep going. Ovaries. What's homologous in the males? Testes, you got it. You're starting to get it. Okay? The vas deferens that go from the epididymis, let's just say the testes, up inside the male body and out to the penis, that is homologous to what in the female? Think about it. If you came up with fallopian tubes, you're right on the money. Okay? When I do the male and female reproductive system, I'll give you some more. But the last one I want to tell you is the labia majora. That's the big outside lips on the females, and the scrotum of a male, homologous. Now you're thinking, well, why are they so big or so much different? When the fetus is developing in the uterus, mom's pumping it, if it's a male, pumping it with testosterone, and that's making those structures grow. You understand? Okay? Enough said. Next, homologous chromosomes and heterologous chromosomes. What the heck is that? Okay, remember those 23 pair you got from mom and the 23 pair you got from dad, the chromatin chromosome? Good? Well, if we line them up, numbers 1, 2, 3, 4, 5, 6, dad's number 1, mom's number 1, dad's number 2, they look kind of like this. I'm going to draw it right next to that silver thing so you can see it. There's number 1. There's number 2, and et cetera, et cetera. They complement each other. Not exactly the same because they're carrying some different genetic information. Different slant on the genetic information. Mom has blue eyes, dad has brown eyes. They're still eye color and they're still at the same spot. Do you get it? Okay. In females, all 23 pair are homologous. All 23 pair are homologous. In males, 1, which is the sex chromosomes, are heterologous. So I'm going to draw it right here. This is the X, this is the Y. There's a section that doesn't match. That makes the 23rd pair heterologous. Got it? So heterologous, homologous chromosomes have nothing to do with homologous analogous structures. Are we good? Okay. Now, let's get into, I'm going to draw. So what I'm going to do is I'm going to move it over this way. I'm going to erase this, and we're going to talk about this column and this one. Got it? Okay, I'm going to pause it so I can get caught up. Okay, we're talking about this column right here, and I'm going to be drawing over here. It says meiosis 1, meiosis 2, tetrad, synapsis, chiasma. Okay, first thing I'm going to do is I'm going to draw chromosome. Yes, it's chromatin. I'm going to draw chromosomes. Remember we talked about a pearl necklace? And I said each pearl is enough genetic information to make a trait. Do you remember that? Okay, so I'm going to draw, I'm either going to make dad red or blue. I'm going to draw chromosome, chromatin, with the genes on it. So it's going to look like a string of pearls. And then with the other color, I'm going to draw mom. And that is what you find in a diploid cell. But this cell is going to undergo meiosis, which means we have to double each one of those. And I'm going to do that. I'm going to show you how it's done. And then I'm going to show you a thing called chiasma, which means the genes can switch back and forth. And that's how we, kind of like shuffling a deck of cards and then dealing them. And when you stop shuffling, you have recombination. And that's how come sometimes you see a kid that looks just like dad or just like mom or a combination of the two. And that's where evolution occurs, only during meiosis and only during prophase in meiosis. Got it? Got it? Prophase and metaphase in a process called synapsis. And that's what's behind me, those words behind me. But I think I'm going to draw it for you so you kind of see what's going on. Ready? Here we go. Yeah, you can see me way over here. I'm drawing the chromatin or chromosomes, and the circles represent traits. Enough genetic information to make a trait. Got it? And let's make this dad. So symbol for dad is that. Okay? Red, we're going to make for mom. Making sure they line up, because they line up, remember? Okay. This is what we have in a diploid cell, cells throughout your body. Even the cells in your gonads are diploid to begin with. Then once they undergo meiosis, which is the only place meiosis happens in the human body, when it undergoes meiosis, things are a little bit different. That's what we're going to talk about. So the first thing we have to do, remember in G1, S, and G2? Remember that in mitosis? What was going on in S? Okay, you're thinking, I don't know, just tell me. No, think about it. Pause this and think about it. Okay, you unpause it, you go, oh, that's right, I remember. DNA synthesis, absolutely correct. We're going to double this. Okay, so the first thing we're going to do is we're going to double dads. You know, this may be the gene for eye color. This might be the ability to make insulin. This might be something else. You understand? Okay. And we're going to do the same thing for mom. This is mom over here. You guys feel like your brains are saturated? Hey, guess what? This is only week 1. Yep, you're the one who said you could do it. All right. Remember in sister chromatids, the sisters are held together by a protein structure called a what? Pause this and think about it. Look in your notes to make sure they're there. I want you to go back to your notes when we were talking about mitosis. And I said the sister chromatids are held together by a protein structure called a what? Okay, look that up. Then turn this back on and here's the answer. Centromere. Centromere. I'm going to draw one right now. So they're all connected by a centromere. Now, in mitosis, we only have the blue, we only have the red. We don't have them together. That centromere starts just up here and down here. But in meiosis, they all hook together. Yes? There's 4 of them. 1, 2, 3, 4. Guess what that's called? You got it, brothers. Tetrad. That is a tetrad. Okay? A tetrad forms in prophase 1. So the first time we in prophase, remember where they're condensing and stuff like that? We form a tetrad. Good? Now, when the tetrad is formed in prophase and the tetrad is moving to the equator, which is in metaphase, metaphase is there. As it's moving there, it's switching genes, but they switch in a line. You understand? They can only switch back because these are bonded together chemically. So when they switch back and forth, they can't go from here to here. They're here. It's always in line. Got it? That switching over is also called crossing over. And another word for that is chiasma. So here I am, chiasma. Another word for crossing over. Got it? It happens from prophase, 1, 2, metaphase, 1. That's when it happens. Only then. The process, now write this down as test, these are test questions, man. The process of going from all over in the nucleus to the equator, the middle, and crossing over. Two things are going on. You're moving and you're crossing over, chiasma. That process is called synapsis. So synapsis is moving from all around the nucleus to the middle and crossing over. Chiasma. Do you understand? Okay. Now let's do a little bit of that crossing over, because this is evolution. I'm concentrating. Okay. Okay. I'm going to re-put in some colors. What I'm doing right now is chiasma. Okay. Now, chiasma's going on during synapsis. Yeah. What happens in anaphase of mitosis? What happened? Spindle fibers started to shorten, and what happened? Do you remember? Pause it and go find out. Don't wait for me to tell you. What I'm trying to do is get you used to going back to your notes so you remember where they're at in your notes. Not just writing them down twice. Well, if I write it down twice, I'll remember. No, you won't. Go back and find it. Pause. Okay, now you came back and you found it. You got a big grin on your face. Okay? You said in anaphase, the spindle fibers shortened and broke the centromere. You were absolutely right. What happened in telophase? Nucleus reformed, right? Just like this. Okay? Now, we once had one cell. How many do we have now? Two. Yes? Yes? But the genome, the genes that were attached on the chromosomes are different now. They scrambled up, aren't they? Yeah. And we no longer have a tetrad. We only have two in there. That's called a dyad. A tetrad, whoops. A tetrad becomes a dyad at the end of meiosis 1. Okay, so the end of meiosis 1, which is telophase, you have two cells. And now you have a dyad. In fact, you have a dyad in anaphase, don't you? Because it broke. but we're going to go ahead and put the nucleus on it before we formally call it a dyad. Okay. Remember we were talking about evolution? Let's just talk about it. We said, you know, we look at families and we look at the offspring, and sometimes look at that. That kid looks just like its father, or that kid looks just like its mother. Yeah? What about if this, I'm going to make it easier for you. Hang on. I don't want you to be confused. Okay, here's my question. What about, I don't want to do that yet. Scratch that. Scratch it. I'm jumping ahead, but I want to make sure I take you through each step. Okay, so we have a dyad and we have a different combination of genes on the chromosome chromatin, correct? A different, what we had all blue here, now we have blue and some red. Here we have blue and mostly blue and a couple of red. Here we have mostly red with some blue. Here we have almost all red. We have a different combination. And that happens in anaphase as soon as that centromere split. And what we have is not called a new combination, it's called recombination. So during anaphase 1, we get a recombination because of synapsis and chiasma. You get that? So synapsis was the moving over at the same time you're going through chiasma. When it breaks, it stops. So whatever you got is called recombination. Got it? And you have a dyad. Okay? Now, then we go through meiosis 2, which is the same process, you know, contracts, bundles up. The only thing is there's not going to be any synapsis or chiasma. That's all controlled by enzymes. There's no enzymes there. Okay, so what happens here is mechanically you move to the middle, metaphase. The spindle fibers pull again, snap the centromere for the second time, and then you don't have any pairs anymore. So I'm going to do that. Okay? We snapped it here. We snapped it here. I'm going to do it in orange so you can see the difference. My question is, this is now a haploid cell. Dyad goes to a haploid cell. N. Remember that? Okay. So for every diploid cell that we start with, after we go through meiosis 1 and then 2, how many haploid cells do we have? That's a test question. There they are. Count. 1, 2, 3, 4. You get it? You get it? So for every diploid cell, we get 4 haploid cells. I'm going to do it by numbers here in a minute. Now, if I put a tail on these and make them a sperm cell, got it? Because it doesn't make any difference if this is going on or mom or dad. This has nothing to do with it. It just said originally where to come from, mom. But this happens to be in dad. Okay? So what about if this strikes the egg? Okay? So mom and dad get together, and that's the one dad releases, that bottom one. What's the kid going to look like? Okay? It's not going to look like mom. It's going to look like grandma, isn't it? You get it? It's going to look just like grandma, except for it's got a mole that dad had. That's the only thing. It looks like that kid was friggin' cloned. Yeah? What about if this one is the one that strikes the egg? Who does it look like? Just about like both grandma and grandpa, huh? Huh? You see? What about, well, you can see what I'm doing. This one's going to look like mostly grandpa with a little bit more of grandma. This one's going to look like grandma and grandpa just about half and half, huh? Do you see? So when you look at somebody and you see that you can see both grandparents, you're going to go, I know how they got that way. Because during chiasma, during synapsis and chiasma, the card that you looked at had characteristics of mostly grandma or of mostly grandpa. You got it? Now, let me take it one step further about evolution. What about if that trait, this one, that was mostly grandma, doesn't survive very good in the world? Okay, now let's take us away from humans and look at other animals, because humans do things a little different because they can change the environment. What about if this is a squirrel? And this is mother's characteristics, and mother wasn't a very good squirrel. She couldn't get much food. She had problems. Do you get where I'm going with this? What's going to happen to the offspring? It's going to get eaten up by a coyote or a fox or a raccoon or something's going to get it. Never going to reproduce. So it removed the weak trait. Yes? What about if it was this one right here? And the traits that it picked up were super strong. Therefore, it can gather food and reproduce better, and you can strengthen the genome of the what they call a gene pool of the species. Do you see how evolution occurs? Now let's look at humans. This is the way it happened with humans. The combination that worked good in us humans was the brain, the cerebrum of the brain, the conscious part of the brain, and the thumb, the opposable thumb. As long as we started going evolutionarily towards that, it took 1 million years, as 2 million years, as we started to move that way, those traits were, those organisms that had that, they were able to reproduce better, and therefore their genetic power was in the gene pool. The population had that. It's called directional evolution. And it happens during synapsis and chiasma in prophase and metaphase 1. Isn't that amazing? Amazing? Okay. You got it? These are haploid cells. Okay? Now, mom's doing the same thing. This guy's wife is doing the same thing with hers. So she produces, for right now, she produces 4. You're not going to find out she only produces 1. But anyway, you got it? Tetrad, synapsis, chiasma crossing over, break, recombination, dyad, do it again, get a haploid. I just showed you how to draw it. And I also threw in a little evolution that way. Pretty good? Pretty good? Okay. What I'm going to do is I'm going to erase everything I've drawn. I'm going to erase this column. I'm going to leave that, and I'm going to put up the female so I can do both of them and finish this up. Okay? Okay. This is male, this is female, and this is development of the fetus. So I'll get to that, but I'm going to do the male first. Okay. Here is a testy. Remember the gonads? And inside the gonads, it's not solid tissue. There's lobules, like cavities. I'll draw them as circles. So the testy is made up of lobules. Do you understand? And coiled up inside the lobules are tubes. And the tubes are called seminiferous tubules. Make sure you know that, it's on the exam. Seminiferous tubules. Got it? Move this over just a little bit this way. Okay, so it's a little more clear, I hope. So we talked about lobules, we talked about seminiferous tubules. Now I'm going to take one of those tubules out and draw it right here. This and that are the same thing. Okay? This is a seminiferous tubule. Seminiferous tubule. Got it? And that tubule is made out of cells. I'll put a dot in here to show that it's got a nucleus. Got it? So they're made out of cells, just like anything else. Now, what's unique, only in males, those cells have a name. They're called spermatogonia. This cell is called a spermatogonia. It has this test question. It has the ability to undergo mitosis and meiosis. So for years and years and years, inside your testis in a lobule, the cell's going through mitosis. All of a sudden, hey man, I need you to become a sex cell. Okay, I need you to become a gamete. So what I'm gonna need you to do is to go through meiosis, 1 and 2. I need you to take your diploid, double it, form a tetrad, break it into a dyad, break it into a haploid, and then I got business for you. Does that make sense? Okay? So what happens is, when it goes through, and I'll change colors for you, when it starts going through meiosis 1, it goes to the next inner layer. This is a tetrad, red tetrad. Green diploid. Got it? And then it's going to go to blue. After a tetrad, what does it go to? What's it go to? Think. Okay, after a tetrad and the centromere splits, it becomes a dyad. What color do I have left? Black. So it goes through it again, and we have a haploid cell. And in this lumen, here's the cell, and it's a sperm cell. See it? Got it? What it does is, all these seminiferous tubules, this is seminiferous tubules, all join inside the testes in kind of like a network. And that's called ret testes. That's inside. Inside, that's terrible inside. It's worse. That's inside the testes. Ret testes. Got it? From there, I'll do it in red so you can see it. All these come together into a storage compartment. These all come together in a storage compartment, and the storage compartment is called epididymis. Red testes, epididymis. And that's where they stay unless they're called upon. I guess you can figure out what it means to be called upon, right? Hey, it's your turn, buddy. Okay, got it? That's male meiosis, called spermatogenesis. The process of doing the tetrad dyad, I already wrote out for you. You got it? Got it? Think if there's anything else I want to tell you. That's pretty much it. Okay, females. Now, if there's anybody out there that thinks a female is not complicated, okay? They don't do it like males. Males hit puberty, and then we go through this stuff, meiosis 1 and 2, a tetrad, a dyad, a haploid, and all the other stuff in the epididymis, red testes. We do it until they put us in the ground or cut our testes off, one of the two. Got it? That's the way we do it. Females, no, they don't do it that way. When they're developing in the uterus in mom, they're going through meiosis 1 they start meiosis 1. Sorry, let me do it that way. They start meiosis 1 and they get to prophase 1 of meiosis 1 and they stop. So in one ovary, you have 200,000 potential eggs. They're not quite there yet. In prophase 1, so they just started to compact and they replicate to a tetrad. Just getting into that part. And then they stop. Got it? The other ovary, same thing. Theoretically, roughly, every female that is born can have 400,000 kids. Idiocracy, huh? Anyway, they can have that. And it stops in prophase 1. Now let me draw an ovary for you. Okay, I'm over here. Looks a lot like that, huh? Because remember, it's homologous characteristics. Now, in the potential egg, I'll make the egg, what's going to be the egg red. They're all in a house. The house is called a follicle. See it right here? The house is called a follicle. Since it has a potential egg in it, it's called an ovarian follicle. Does that make sense? So at birth, you ladies have 400 ovarian follicles with ova in it. They're not a complete ova. They're the makings of an ova. Do you get it? Okay. Okay. And then once you're born, it stops. And it stops until you get to puberty, and then one is selected. Hey, you're going to be an egg that's going to be ovulated. You could end up getting pregnant and having a beautiful child. And if you're 12 years old and that happens, I'm going to come talk to you. Okay? All right. So what I'm going to do over here is, instead of showing you 400 or 200,000 of these, I'm going to draw one in succession as it is awakened at puberty. And it says, hey man, it's your turn. The follicle starts to swell. And it swells until it gets to a point where it's going to burst. Do you understand? That's the follicle. Two things are going to go on here. The follicle is going to change. The egg is actually undergoing metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, and then it stops again. But the egg's not going to change in size. It's going to look just the same. But we know that inside, remember how I drew it with the pearls? Remember that? It's going on. So when you're awoken, hey man, you're going to be an egg. Then it says, okay, what do I have to do? Oh yeah, I'm a tetrad. I have to move to the equator, synapsis, and I'm going to do some chiasma. I'm going to do some crossing over. And then in anaphase 1, I'm going to split. All the crossing over is done. I go to telophase, you wrap me up, you make me a dyad, and then I'm going to go through it again. And I get to prophase, and then I get to metaphase 2, and you say, I'm tired of this. I don't want to play no more. Okay? And what happens is you go from an ovarian follicle with an oocyte in it, and then primary, this one is primary, okay? Synapsis, in the primary synapsis, chiasma, okay? And it goes until telophase, I'll put TELO, 1, and then it goes into secondary. Put 2N, that's why I do it, secondary. And that's a dyad. And then it lines up at the equator. Here's the egg. I mean, the cell lines up at the equator. The secondary follicle, secondary ova or oocyte, because they match. Secondary follicle, secondary oocyte. Primary follicle, primary oocyte. Got it? They match. And that's where it goes. It stays, it goes, this is a test question. Metaphase 2. That's what females ovulate. It's a dyad. It hasn't split yet. It's not a haploid cell. That's what she ovulates. Got it? Now, just before ovulation, this is where we're at. Just before ovulation, that follicle is so big. It's like when you blow up a balloon and you know that the next breath is going to pop, right? Well, the next growth of this follicle, it pops. And when it does, just before it pops, this is called Graafian. Graafian. It's called a Graafian follicle. And it contains a potential, okay, a potential ova in metaphase 2. You got it? Get this really squared away in your brain because I'm going to ask you a couple of test questions on it. Okay? If you can't figure this out, you're going, oh, try it, read over your notes, read over the book, and if it still doesn't work, go to Brooke on SI or tutoring, and she'll go over it again. If that doesn't work, you get a hold of me, and I will go over it, because remember, I have an office hour at noon. Okay, that's secondary, Graafian, and when it pops, the Graafian pops, I'll make it look yellow. Well, let me do that again. Now I got room to do one more cell. Okay, it's Graafian. Then it pops. And the egg comes out. That's in metaphase 2. The egg comes out. There's a name for that. It's called ovulation. Okay, here's where I'm at. Ovulation. Now the follicle is empty. Yes? Follicle is empty. Latin for body is corpus. Latin for empty is luteum. Next, corpus luteum. That's what this thing is called. It's called corpus luteum. Empty body. And then it's all under control of hormones. It starts to shrivel up. And then it's called corpus albicans. So, ladies, however many times you have ovulated, you have a corpus albicans, a little shriveled up little scar in your ovary. Yep. Yep. Okay, next step. Let's talk about fertilization. I'm going to start. Okay, here we go. We talked about ovulation. Remember the Graafian follicle, swell, burst, out comes metaphase 2. We call it an egg, but it's not technically an egg because it's not haploid yet. Okay, and then as it does, it starts its way down the fallopian tubes. And I have a video I'm going to have you watch on this. Now, if this is the egg, I'm going to put ova, and this is the sperm. Happy face. The sperm does not go through the phospholipid bilayer. There's an extra covering around here. And there's a kind of like a mucus around here. You'll see it in the video. And as the sperm comes to here, it has to neutralize this blue ring, which is called corona radiata. So this blue ring is called corona radiata. Got it? And it does that with an enzyme called acrosome. So I'll do that in green right here. A little acrosome, and it goes down here, and if it has the right spot at the right time, it will allow an opening to occur. And then in comes the sperm. This is the phospholipid bilayer. As it comes through the phospholipid, here's the nucleus. As it comes through the phospholipid bilayer, it drops the flagellum, that's the tail. And the only thing you have is the outside phospholipid bilayer with the 23 chromosomes. Okay, so I'm going to get rid of the ova, and I'm going to I'll put them in different colors so you see it. This is the nucleus. This is the nucleus of mom with 23. Here's the nucleus of dad with 23. They merge, and when they merge, you get 46. And then you're back to a diploid cell. What I just showed you is fertilization. Corona radiata is the outside covering. Between the corona radiata and the phospholipid bilayer of the mom's egg is called zona pellucida. Got proteins in it and stuff. So the sperm has to get through corona radiata using acrosome. Then it goes through the zona pellucida and through the phospholipid bilayer, and then we have fusion of nuclei, which equals fertilization. Okay, fertilization. One egg, one sperm forms one cell called a zygote. Once a zygote is formed, we're right back to mitosis. Got it? And then you go to a morula. We're going to do this when we do the lab on, just a minute, the lab on mitosis. We'll talk about a morula. A morula, after the zygote goes through a whole bunch of mitotic divisions, and you have a solid mass of cells. That's called a morula. As it continues to divide, cleavage, it hollows out. A mass of cells that's hollow is called a blastula. And then as it gets big, then it starts to invagine. It kind of makes like a figure eight. And that's the first process, organogenesis, that forms, which is the digestive tract. This being the mouth, this being the anus. And then it continues with the germ layers. Good? Okay. Organogenesis. Now, I have a video to go through this. But before we do that, I want to do one other thing. I'm going to do some math for you. Okay? Let me erase this. Okay? Now, I'm going to talk while I'm doing this because the math is just following chromatin or chromosomes. I'm just going to call it chromosomes right now. Remember those pearl necklaces we were talking about? Okay, this math is just forming those. Okay? Not forming those, following those. Sorry, my mind drifted. Okay? Now, we're going to be revisiting in this math thing that we're going to do. We're going to revisit tetrad, dyad, diploid cell, haploid cell, meiosis 1, meiosis 2, and mitosis. Okay? So the first thing I'm going to draw for you is mitosis. Okay? Just using numbers in a nucleus. And the numbers are the number of necklaces, chromatin or chromosomes. Okay, you start with 46, diploid cell. Whoops. You put up above it so you can see what the heck we're talking about. Here's mitosis. We good? Here's meiosis. Okay, you start with the cell, a diploid cell in both cases. 46. Remember in the male, the diploid cell was spermatogonia. Okay, Then we go through S phase. What would happen in S phase? Come on. What happened in S phase? S synthesis. DNA replicated. So the doubles. 46 and 46 is 92. Still one cell with 92. Still one cell with 92. Now you'd say, well, they're absolutely identical. No. This one is 46 sister chromatids held together by a centromere. So they're doubles. They're not dyads, they're just doubles. A replicated DNA, of mom, 23, and dad, 23, but they're hooked together. The moms are hooked together and the dads are hooked together in doubles, not in fours. Got it? Here, there's a tetrad. So let me write that down for you so you can see it. So here, we have 46 sisters. They call them sister chromatids. Here, we have 23 tetrads. 4 times 23 is 92. 2 times 46, 92. Do you see it? So when you look at it mathematically, it looks like they're the same. But you know, inside the nucleus, they're not. Okay. Then, we go through mitosis, prophase, metaphase, anaphase, telophase, and we get this. Cloned cells. I'll put cloned. 2 cloned cells. And we call that cell replication. Here, I'm going to use this a different color. We split, we get 46, 46, so we get 2 cells of 46, and this is meiosis 1. Okay, and this is a dyad. Okay, another color. Then this splits. We get 23, 23, 23, 23. These are haploid. N. Got it? Doesn't look like this, does it? So the result of one cell going through mitosis is 2 identical cells. The result of meiosis of one diploid cell results in 4 haploid cells. Put a little tail on them. Guess what we have? Sperm. Okay? Let's look at a female. Gotta love them. Okay, this is a male. Here's a female. 46. Potential ova in an ovarian follicle at birth. S phase, 92. Okay. Prophase 1, go to sleep for 12 years until puberty. Okay. Then if it wakes up, we have unequal cytokinesis. Kinesis, movement of the cytoplasm, cytokinesis. So what happens instead of the cytoplasm, this is outside the nucleus. Instead of the cytoplasm, remember the cytosol and that kind of stuff? Instead of it dividing equal, one gets it all, the other one gets nothing. Okay? That's why an oocyte is 120 micrometers in size, largest cell in the human body, largest cell in the human body, largest cell in the human body. So what we get is, we get 46 and 46. You see? Got all the cytoplasm. This is a tetrad. Let me do the blue again. This is a tetrad. This is This is a dyad. Okay. This is a tetrad. Okay. I just didn't have them on the same thing. Then we divide again. Guess what? Happens again. And this time, the 46 does 23 and 23. And this is the ova. So, watch. This is your test question. The result of spermatogenesis is 4 equal haploid cells. The result of oogenesis is 1 egg or cell, and 2, we call these cell bodies. Got it? Because this one, this 46, goes here. This 23 goes here. So what that 23 looks like is this with one with 46 and one with 23. So you get 1 egg and 2 cell bodies. Yes? Son of a gun. Okay. Okay. That's it for meiosis. I want you to go onto what's the one that has all the videos? Just a minute. My mind went blank. Amazing, I just remembered. Go on YouTube, and I want you to type in, see if it'll do it. Video clip, meiosis, the continuation of life. It's 11 minutes and 49 seconds. I want you to do this because it is a complete review of 2 things, mitosis and meiosis. It is excellent, excellent video. And you can pause it and guess what it's doing. But you're going to see the egg tumbling down, that egg, tumbling down the fallopian tubes. You're going to see the cilia waving, and then you're going to see the sperm are trying to find it. And then when it does, you see fertilization. And you're going to see the zona pellucida. You're going to see corona radiata. You're going to see all those things we talked about. Got it? Got it? And then it's going to stop. And then it's going to, because it's going to start mitosis, you're going to see a morula, a blastula, and a little bit of gastrula, and you're going to see the placenta form. And then they're going to stop, and they're going to go back through meiosis. And you're going to see forming of the tetrad. You're going to see prophase 1, metaphase 1, and so on and so forth. Excellent, excellent, excellent tool. Okay? Now, that's it for the meiosis, which is part of chapter 3. And the next time we talk, I'm going to do integument for lecture. Integument, which is, no, I'm telling you back, it's going to be histology. Histology, and then integument, and then we have an exam Monday at noon. Okay? Now I have to I'm going to go through and do the labs for you and lay them out and stand up here and explain to you why we're doing things a certain way. Okay? Have a good day.